Hello, ladies and gentlemen. Welcome to chapter 25. Okay, let's get started. When a cell is not dividing, its nucleus is occupied by chromatin. DNA, deoxyribonucleic acid, is twisted around something known as histones. This is chromatin. These little gumball looking things are histones. So when your DNA is wound around these guys, it's called chromatin. So here's your DNA. It winds around the histones and becomes chromatin, and then it becomes a chromosome. Why does it do this, you ask? DNA is wrapped around the histones to regulate gene expression. This is an actual question on sapling learning. So write it down because it's a question on sapling. Let's think about that for a second. When you have your DNA wrapped around this guy, do you think it's going to be real easy for mRNA to get right in there and transcribe some mRNA? It's not, because it's all wrapped up in here. So the way in which your chromatin winds around these things, this is what regulates gene expression. What gets expressed, that's exposed up here, and what your mRNA can't get to. Okay. During cell division, chromatin organizes itself into chromosomes. These should look familiar. We can karyotype you, which is look at all of your chromosomes when they are ready to duplicate. They are called chromosomes. Each chromosome contains a different DNA molecule. The DNA is duplicated so that each new cell receives a complement copy. What does that mean? Here's your old DNA. It splits, and then this yellow one is the new copy. Do you see the yellow one? This yellow one over here is the new copy, and it stays together, so you'll get a complete DNA with one old and one new, and another copy here with one old and one new. Okay. Each DNA molecule is composed of genes. This is very confusing for people. What is a gene? A gene is a chunk of DNA that codes for something. So it would be the gene for dark hair, or the gene for sickle cell anemia, or the gene for invisibility. That's coming. We're going to find that gene. Just give us time. So this is the DNA, this is the chromosome, and the segment right here, it's actually probably longer than that, is a gene. It's something that codes for a specific thing. It codes for a specific protein that does a specific thing. And that's why we're calling it a gene, because it codes for a specific protein. Nucleic acids are polymers known as polynucleotides. It's just more than one nucleotide. It's pretty easy if you think about it, just polynucleotides. Nucleic acids are polynucleotides. Each nucleotide has 3 parts. Let's look. A 5-membered monosaccharide. So this guy here is a monosaccharide. It's got 5 carbons. 1, 2, 3, 4, 5. This is not a carbon. So it goes 1, 2, 3, 4, 5. That is our 5-membered monosaccharide. Then there is a nitrogenous base right here, and then there is a phosphate group. Okay, does everyone see the parts of a nucleotide? Okay. The sugars. Now, in RNA, you have ribonucleic acid. In DNA, you have deoxyribonucleic acid. What is the difference? Look at this guy. This guy has 1, 2, 3, 4, 5 carbons, but it's missing this oxygen, this alcohol here. This one has the same 1, 2, 3, 4, 5 carbons, but it has this. It really will have 3 alcohols, one here, and one here. This one is missing one, so it's deoxy, 2 oxygens, ribose. Does everyone see that? Okay. I'm pretending they're here, so I'm asking you questions. We're having a little dialogue. I can hear you. The 5 nitrogenous bases found in DNA are purines and pyrimidines. I'll show you pictures of those in just a minute. A purine always goes with a pyrimidine. A goes with T or U. G goes with C. A purine goes with a pyrimidine. They're made for each other. In addition to differing in the sugars, they contain RNA and DNA. Thymine is present only in DNA. Uracil is present only in RNA. So if A goes with U in RNA, A goes with T in DNA. Here's the promised picture. Here's our purine. So there's 2 purines, adenine and guanine. See how they have 2 rings? This is our pyrimidine here. It's just one ring. 1, 2, 3, 4, 5, 6. And it's cysteine, which goes together with guanine. Do you see how the puzzle fits together? You have to have a 2-membered ring with a 1-membered ring. C goes with G. Then you have thymine or uracil, depending on if it's DNA or RNA, and it goes with adenine. Does that make sense? Okay. Let's move on to nucleosides and nucleotides. A nucleoside does not have phosphate. A nucleotide does have phosphate. This is a nucleoside. This is a nucleotide. The difference is this little phosphate ring right here. A nucleoside plus phosphate equals a nucleotide. It just has a phosphate ring. We just said that. And there's some of them there if you want to see what they are. In summary, a nucleoside is a sugar and a base. A nucleotide has the phosphate group. This DNA and RNA and the differences, this will show up probably on a test, and I think it even shows up on Sapling Learning somewhere. DNA is a polymer of deoxyribose. RNA is ribonucleotides. The sugar is 2-deoxy-D-ribose. The sugar is just D-ribose. The bases are adenine, guanine, cytosine, and thymine. The bases here are uracil. This versus this. That will show up. The nucleotides in DNA and RNA are connected by phosphate diester linkages. We talked about this. Here they are, a 3,5 phosphodiester linkage. It's got this little phosphate group here. That is a phosphate diester bond. It is between the OH group, the 3' sugar, and the phosphate group. Let's count our carbons. 1, 2, 3. There was an OH there, if you remember what it looked like. Remember, there was an OH there. And the 5'. 1, 2, 3, 4, 5. Between the 3rd carbon and the 5th carbon. That is where that phosphodiester bond is. The structure and function of nucleic acids depends on the sequence. We talked about this, the beads on a chain. The sequence is the most important thing, and the folding is going to include the sequence. The sequence of nucleotides in a nucleic acid chain is read by starting at the 5' end. So you have this long chain, and you will say to yourself, I don't know where to start. Where's the head? Where's the beginning? I have no idea. You automatically orient yourself to the 5' end. How do you know if it's the 5' end? Because it has this phosphate group on it. 1, 2, 3, 4, 5. The 5th carbon, which has the phosphate, this is the head. This is where you start. This is the way you read everything from 5' to 3'. Let's talk about Chargaff. Chargaff's rule. Chargaff's rule says A goes with T, C goes with G. This is his basic rule. He figured out through analysis, this handsome guy here with a little bow tie, A goes with T, C goes with G. That's his basic. You can't read these numbers. They're really fuzzy. But when he analyzed DNA, he found that there was always exactly the same amount of adenine and thymine. Always the same amount of cytosine and guanine. So he said to himself, I bet that's how they link up. That's how they connect. A is always connected to a T. T is always connected to an A. And that's how they replicate each other. So amounts of adenine and thymine were always equal. And so he realized that this was the way that things came together. The properties of A goes with T and C goes with G is Chargaff's rule. The sugar phosphate backbone is on the outside of the right-handed double helix. The heterocyclic bases are on the inside. So a base on one strand points directly towards the other. This is huge. Let's look at this for a second. So these phosphate, remember our little phosphate? Remember our 5' end? Where's the head? Here's the head because here's the phosphate group, 5' end. Where's the head here? It's upside down. Here's the 5' head. So the phosphate groups are on the outside, making up this part of the ladder when you look at it, this part of the ladder. And these guys, your A and your T, connect in here, in the center. The C and G have 3 bonds. The A and T have 2 bonds. Oh, I have to go because it can't be more than 10 minutes. I'll see you in a minute.